There is a muscle in your body that you have never consciously contracted. You have never seen it in a mirror. No personal trainer has ever pointed at it on a chart and said, "Give me 3 sets of 12." It does not appear in any gym routine. It does not respond to dumbbells or resistance bands or any movement you would recognize as exercise. And yet this muscle, or more precisely this network of muscle tissue wrapped around structures you cannot voluntarily control, is determining how fast your cardiovascular system ages, how well your organs receive blood, how efficiently waste is removed from your tissues, and whether the biological clock ticking inside every one of your cells runs at the pace of a 50-year-old or an 80-year-old. The muscle is your vascular smooth muscle, the layer of contractile cells that lines every artery and arteriole in your body. And the reason you have never trained it is that you cannot flex it, you cannot isolate it, you cannot feel it working. But it is working right now. It contracted and relaxed approximately 70 times in the last minute, responding to every heartbeat, adjusting the diameter of your blood vessels, regulating the pressure wave that carries oxygen to your brain, your kidneys, your muscles, your skin, and it is aging, faster than it should, because the inputs it needs to maintain its function are inputs that modern life has systematically removed. Let me explain what this tissue actually is, because the physics of it matters. Your arteries are not pipes. This is the most common misconception in cardiovascular medicine, and it leads people to think about heart disease completely wrong. A pipe is rigid. It has a fixed diameter, fluid flows through it passively. Your arteries are nothing like this. They are dynamic, living, muscular tubes that actively participate in every heartbeat. The wall of a major artery, like your aorta, has 3 layers. The innermost layer, the tunica intima, is a single-cell thick endothelium that contacts the blood directly. The outermost layer, the tunica adventitia, is connective tissue that anchors the vessel to surrounding structures. But the middle layer, the tunica media, is where the machinery lives. This is smooth muscle. Layers of spindle-shaped cells arranged circumferentially around the vessel, embedded in a matrix of elastin and collagen fibers. When these muscle cells contract, the artery narrows. When they relax, the artery dilates. The balance between contraction and relaxation at any given moment determines your blood pressure, your organ perfusion, your tissue oxygenation, and over decades, the structural integrity of the vessel wall itself. Here is the physics that makes this remarkable. Your heart ejects blood in pulses. Each contraction of the left ventricle sends a pressure wave into the aorta at roughly 120 millimeters of mercury. If your arteries were rigid pipes, that pressure wave would slam into your organs at full force 70 times per minute. Your brain would experience hydraulic hammering with every heartbeat. Your kidneys would be destroyed within days. But your arteries are not rigid. The smooth muscle and elastin in the arterial wall absorb the pressure wave. The aorta expands with each heartbeat, storing the kinetic energy of the pulse in the elastic stretch of its wall, and then recoils between beats, pushing the blood forward during diastole. This is called the Windkessel function, named after the German word for air chamber, because it works exactly like the air chamber in an old fire engine pump that converts pulsatile flow into continuous flow. Your vascular smooth muscle is half of this system. The elastin provides passive recoil. The smooth muscle provides active tone, adjustable resistance, and moment-to-moment regulation of how much the wall gives and how much it pushes back. Without functioning smooth muscle, the Windkessel fails. The pressure wave travels faster. It reflects off branch points and returns to the heart before the next beat is complete. The heart has to pump against its own reflected wave. This is arterial stiffness, and it is the single strongest predictor of cardiovascular death in people over 50, stronger than cholesterol, stronger than blood pressure measured at the arm, stronger than smoking history. The speed at which the pressure wave travels through your arteries, called pulse wave velocity, is a direct measurement of how stiff your vascular smooth muscle has become. And it increases with age at a rate that is not fixed, it is modifiable. The rate depends on what you do. Now I need to tell you what makes vascular smooth muscle different from the skeletal muscle you train at the gym, because this difference is why nobody talks about it, and why it degrades silently for decades before the consequences become catastrophic. Skeletal muscle is voluntary. It is innervated by somatic motor neurons that you consciously control. You decide to lift your arm, and the motor cortex sends a signal down the corticospinal tract to the alpha motor neurons in your spinal cord, and the biceps contracts. You feel it. You see it. You can make it stronger by repeatedly demanding more of it. Vascular smooth muscle is involuntary. It is controlled by the autonomic nervous system, by circulating hormones, by local chemical signals from the endothelium, by the partial pressure of oxygen and carbon dioxide in the surrounding tissue, and by the mechanical forces of blood flow itself. You cannot decide to dilate your femoral artery. You cannot consciously contract the smooth muscle in your renal arterioles. The system operates below the level of awareness, regulated by feedback loops that evolved over millions of years to maintain homeostasis without requiring your attention. But here is what most people do not understand. Just because you cannot voluntarily control this muscle does not mean you cannot train it. You can. The training stimulus is not a conscious contraction. The training stimulus is hemodynamic stress, the physical forces that blood flow exerts on the vessel wall, and the most important of these forces is called shear stress. Shear stress is the frictional force of blood flowing across the endothelial surface. When blood flows faster, shear stress increases. When shear stress increases, the endothelial cells detect it through mechanosensitive proteins on their surface and respond by producing nitric oxide. Nitric oxide diffuses into the smooth muscle layer and causes relaxation. The vessel dilates. Blood flow increases further, and the smooth muscle cells, in response to this repeated cycle of stretch and relaxation, maintain their contractile phenotype. They stay healthy. They stay responsive. They stay young. The problem is what happens when shear stress is chronically low, when you sit for 8 hours, when you do not move enough to raise your heart rate, when blood flow through your peripheral arteries remains at baseline for days and weeks and months. The endothelium produces less nitric oxide. The smooth muscle receives less relaxation signal. It begins to shift from a contractile phenotype to what vascular biologists call a synthetic phenotype. This is not a metaphor. It is a measurable cellular transformation. The smooth muscle cells stop expressing the proteins that allow them to contract and relax efficiently. They start expressing proteins associated with proliferation, migration, and extracellular matrix production. They become stiffer. They produce more collagen. They produce less elastin. The vessel wall thickens. The compliance decreases. The Wind-Kessel function degrades. The pulse wave velocity increases. You are aging, not because time passed, because the mechanical stimulus that maintained your vascular smooth muscle in its youthful state was removed. Daniel Green at the University of Western Australia has spent 2 decades measuring this with extraordinary precision. His laboratory uses flow-mediated dilation, a technique where you occlude blood flow to the forearm with a cuff, release it, and measure how much the brachial artery dilates in response to the sudden increase in shear stress. The dilation is entirely dependent on endothelial nitric oxide production and smooth muscle responsiveness. It is a direct functional test of your vascular smooth muscle health. Green's data shows that flow-mediated dilation decreases with age at a rate of roughly 0.5% per year after age 25 in sedentary individuals. By 65, a sedentary person has lost roughly 20% of their flow-mediated dilation capacity compared to their peak. But here is the finding that changes everything. In people who maintain regular aerobic exercise throughout their lives, the decline is cut by more than half. 70-year-old endurance athletes show flow-mediated dilation values comparable to sedentary 40-year-olds. Their vascular smooth muscle is 30 years younger than their birth certificate. The exercise did not stop time. It maintained the mechanical stimulus that keeps smooth muscle in its contractile phenotype. The shear stress from regular elevated blood flow told the endothelium to keep producing nitric oxide. The nitric oxide told the smooth muscle to keep relaxing. The repeated relaxation told the smooth muscle cells to keep expressing contractile proteins. The signal chain remained intact. The muscle stayed young because it kept receiving the stimulus it was designed to receive. But Green found something else that is perhaps more important for anyone who has not been exercising for decades. The vascular smooth muscle responds to training at any age. His studies in previously sedentary older adults showed significant improvements in flow-mediated dilation after just 8 to 12 weeks of regular aerobic exercise. The smooth muscle cells can shift back toward the contractile phenotype. The endothelium can upregulate nitric oxide synthase. The compliance can improve. The pulse wave velocity can decrease. The biological age of your arteries is not fixed by your chronological age. It is responsive to the mechanical environment you create through movement. The muscle you never trained can still be trained. The factory can be restarted. Now I need to explain why this matters more than almost anything else in aging medicine, and the explanation requires understanding what arterial stiffness actually does to your organs. Your brain receives roughly 15% of your cardiac output. It is the most metabolically demanding organ in your body relative to its size. It requires continuous, steady blood flow, not pulsatile hammering, continuous perfusion. The Wind-Kessel function of your large arteries is what converts the pulsatile output of your heart into the steady flow your brain needs. When your arteries stiffen, when the smooth muscle loses its compliance, the pulse wave is no longer absorbed. It transmits directly into the small vessels of the brain. The cerebral microvasculature, tiny arterioles with walls only 1 or 2 cells thick, receives pressure pulses they were never designed to handle. Over years, this pulsatile damage causes microbleeds, white matter lesions, and progressive destruction of the small vessel network that supplies the deep brain structures. This is called cerebral small vessel disease. It is visible on MRI as white matter hyperintensities, bright spots scattered through the brain tissue that represent areas of chronic ischemic damage, and it correlates directly with pulse wave velocity. The stiffer your arteries, the more pulsatile energy reaches your brain, the more white matter damage accumulates, the faster your cognitive function declines. The Rotterdam study, a massive longitudinal cohort that has followed thousands of older adults for decades, showed that arterial stiffness measured at baseline predicted cognitive decline and dementia risk over the following 10 to 15 years, independent of blood pressure, cholesterol, diabetes, and every other traditional risk factor. The mechanism is purely mechanical. Stiff arteries transmit damaging pressure waves to the brain. The brain accumulates microvascular damage, cognition declines. The vascular smooth muscle that you never trained, that you never thought about, that no one ever told you was important, is protecting your brain from mechanical destruction with every single heartbeat. When it fails, your brain pays the price. Your kidneys face the same problem. They filter your entire blood volume roughly 40 times per day. They are perfused at enormously high flow rates through a specialized vascular bed that is exquisitely sensitive to pressure. The glomerular capillaries, where filtration occurs, operate within a narrow pressure window. Too little pressure and filtration stops. Too much pressure and the delicate filtration membrane is damaged. Arterial stiffness increases the pulsatile load on the renal vasculature exactly as it does in the brain. The result is progressive glomerular damage, declining filtration rate, and the slow march toward chronic kidney disease that affects roughly 40% of people over 70. The connection between sedentary behavior and kidney decline runs through the same vascular smooth muscle pathway. Less movement means less shear stress, means less nitric oxide, means stiffer arteries, means more pulsatile damage to the kidneys, means declining filtration, means accumulating waste products, means accelerated aging of every tissue in the body. There is a concept in vascular biology called vascular age versus chronological age, and the gap between them is determined almost entirely by the functional state of your smooth muscle. A 55-year-old who has been sedentary for 20 years may have arteries that function like those of a 70-year-old. A 70-year-old who has maintained regular aerobic activity may have arteries that function like those of a 50-year-old. The difference is 20 years of biological age in the same organ system. And because the arterial system supplies every other organ, vascular age propagates. Your brain ages at the rate your arteries age. Your kidneys age at the rate your arteries age. Your heart ages at the rate your arteries age because it must pump against the resistance your stiff arteries create. Left ventricular hypertrophy, the thickening of the heart muscle that precedes heart failure, is a direct adaptation to increased arterial stiffness. The heart gets bigger because it has to work harder, because the arteries lost their compliance, because the smooth muscle shifted phenotype, because the shear stress disappeared, because you stopped moving. Let me tell you about the endothelium because it is the command center for everything I have described, and it is the tissue that translates your behavior into vascular smooth muscle function. The endothelium is a single layer of cells lining the interior of every blood vessel in your body. If you spread it out flat, it would cover roughly 4 to 7 tennis courts. It weighs about 1 kilogram. It is an organ, the largest organ by surface area in your body, and it is a signaling organ. It detects the mechanical and chemical environment of the blood and translates that information into instructions for the smooth muscle layer beneath it. The primary signal it produces is nitric oxide, synthesized by an enzyme called endothelial nitric oxide synthase, eNOS. When shear stress is adequate, eNOS is active. Nitric oxide production is high. The smooth muscle relaxes. The vessel dilates. Anti-inflammatory pathways are activated. Platelet aggregation is inhibited, smooth muscle proliferation is suppressed, the vessel stays healthy. When shear stress is chronically low, eNOS expression decreases. Nitric oxide production falls. The smooth muscle contracts. The vessel narrows. Inflammatory pathways activate. Oxidative stress increases. The smooth muscle begins its phenotypic shift toward the synthetic state. The vessel stiffens. Atherosclerotic processes accelerate. The endothelium is the translator between your movement patterns and your vascular age. Every time you elevate your heart rate, every time blood flows faster through your arteries, the endothelium detects the increased shear stress and responds by producing the molecule that keeps your smooth muscle young. Every hour you sit still, the signal weakens. There is a dose-response relationship here that has been carefully measured. The minimum effective dose for maintaining endothelial function and vascular smooth muscle health appears to be approximately 150 minutes per week of moderate intensity aerobic activity. This is not coincidence. This is the number that appears in every cardiovascular guideline from every major health organization in the world. It was not chosen arbitrarily. It emerged from decades of epidemiological data showing that this threshold separates populations with preserved vascular function from populations with accelerated vascular aging. But the relationship does not plateau at 150 minutes. Additional activity provides additional benefit up to roughly 300 to 400 minutes per week, beyond which the returns diminish but do not reverse. The smooth muscle responds to cumulative shear stress exposure. More time with elevated blood flow means more time with high nitric oxide production, means more time the smooth muscle spends in its relaxed, contractile phenotype. But here is what makes this particularly relevant if you are over 60. The type of exercise matters less than you think. The critical variable is not intensity, it is consistency of the hemodynamic stimulus. Walking briskly enough to elevate your heart rate to 60 to 70% of maximum provides sufficient shear stress to maintain endothelial function. You do not need to run. You do not need to cycle at race pace. You need blood to flow faster than resting baseline through your arteries for a sustained period, regularly. 30 minutes of brisk walking 5 days per week delivers the mechanical stimulus. Your vascular smooth muscle needs to maintain its contractile phenotype. The smooth muscle does not know whether you are walking or running or swimming or cycling. It knows whether shear stress is present or absent. It responds to the physics of flow, not the sociology of exercise. There is another input to vascular smooth muscle function that operates through a completely different mechanism, and it explains why breathing patterns matter in ways that seem almost absurd until you understand the chemistry. Carbon dioxide, the gas you exhale with every breath. It is not merely a waste product, it is a potent vasodilator. Carbon dioxide dissolved in your blood as carbonic acid directly relaxes vascular smooth muscle through pH-dependent mechanisms. When tissue carbon dioxide levels rise, local arterioles dilate to increase blood flow and wash the carbon dioxide away. This is called flow metabolism coupling, and it is the primary mechanism by which your brain regulates its own blood supply. Active brain regions produce more carbon dioxide. Local vessels dilate. More blood arrives. The neurovascular coupling that keeps your brain functioning depends on vascular smooth muscle responding to carbon dioxide. Here is where this becomes practical. Chronic hyperventilation, breathing more than your metabolic rate requires, lowers your blood carbon dioxide levels below the optimal range. The medical term is hypocapnia, and it is extraordinarily common in sedentary, stressed, anxious populations. When carbon dioxide drops, vascular smooth muscle contracts. Cerebral blood flow decreases by roughly 2% for every 1 millimeter of mercury drop in arterial carbon dioxide. A person who chronically overbreathes, taking 15 to 20 breaths per minute at rest instead of the optimal 8 to 12, may be running their cerebral blood flow 10 to 15% below what it should be. Chronically, for years, the vascular smooth muscle in their brain is in a state of sustained partial contraction because the carbon dioxide signal that should keep it relaxed is being blown off through excessive ventilation. Konstantin Buteyko, the Russian physician who developed breathing retraining methods in the 1950s, understood this relationship decades before Western medicine acknowledged it. His clinical observation was that patients who breathed less, who tolerated higher carbon dioxide levels, had better circulation, lower blood pressure, and fewer symptoms of chronic disease. The mechanism is vascular smooth muscle relaxation driven by carbon dioxide. Slow breathing, 5 to 6 breaths per minute, maintains higher alveolar carbon dioxide, which maintains higher arterial carbon dioxide, which maintains vascular smooth muscle in a more relaxed state, which maintains better organ perfusion. The breathing rate you maintain throughout your day is a continuous input to your vascular smooth muscle tone. Fast, shallow breathing contracts it. Slow, deep breathing relaxes it. The muscle you never trained responds to the breath you never thought about. There is a third input that I need to describe because it operates on a timescale that makes it invisible, but its effects are devastating. Chronic psychological stress activates the sympathetic nervous system. The sympathetic nervous system releases norepinephrine at the neuroeffector junctions embedded in the smooth muscle layer of every artery in your body. Norepinephrine binds alpha-1 adrenergic receptors on the smooth muscle cells and causes contraction. This is the fight-or-flight vasoconstriction that redirects blood from your gut and skin to your muscles when you face a threat. It is supposed to last minutes. In modern life, for many people, it lasts years. Chronic sympathetic activation means chronic norepinephrine release, means chronic smooth muscle contraction, means chronic elevation of vascular tone, means chronic hypertension, means accelerated arterial stiffening, means accelerated vascular aging. The stress you carry in your body is not a metaphor. It is norepinephrine binding alpha-1 receptors on your vascular smooth muscle and holding it in contraction for years. The muscle ages faster because it never relaxes. It never receives the signal to return to its resting state. The parasympathetic nervous system, the rest and digest branch, releases acetylcholine that stimulates endothelial nitric oxide production and promotes smooth muscle relaxation. But, in a chronically stressed state, parasympathetic tone is suppressed. The relaxation signal is absent. The contraction signal is continuous. The smooth muscle remodels around its contracted state. The vessel wall thickens. Compliance decreases. The damage accumulates silently for decades until a stroke or a heart attack or a diagnosis of heart failure reveals what has been happening in the dark. This is why meditation, yoga, tai chi, and other practices that activate the parasympathetic nervous system have measurable effects on blood pressure and arterial stiffness. They are not mystical. They are not placebo. They reduce sympathetic outflow to the vascular smooth muscle. They allow the muscle to relax. They restore the balance between contraction and relaxation that the muscle needs to maintain its youthful phenotype. The mechanism is neurochemical. Norepinephrine goes down, nitric oxide goes up, the smooth muscle relaxes, the vessel dilates, blood pressure drops. The effect is measurable with a blood pressure cuff within minutes of beginning a relaxation practice. The long-term effect, measured over months and years of regular practice, is reduced arterial stiffness and reduced pulse wave velocity. The muscle you never trained responds to the stress you never resolved. Let me bring this together because the picture is now complete, and it is both terrifying and hopeful. Your vascular smooth muscle is the tissue that determines how fast your cardiovascular system ages. It controls your blood pressure, your organ perfusion, your brain health, your kidney function, and the workload on your heart. It responds to three primary inputs: hemodynamic shear stress from physical movement, carbon dioxide levels from your breathing pattern, and autonomic nervous system balance from your psychological state. All three inputs have been degraded by modern life. We move less than any generation in human history. We breathe faster and shallower due to chronic stress and sedentary posture. And we maintain sympathetic activation at levels our ancestors experienced only during acute threats. The result is a population whose vascular smooth muscle is aging 20 to 30 years ahead of schedule, whose arteries at 55 function like those of a 75-year-old, whose brains are accumulating white matter damage from pulsatile pressure that should have been absorbed by compliant vessel walls, whose kidneys are declining because the filtration membrane is being hammered by pressure waves that a healthy smooth muscle layer would have dampened. But the hopeful part is this: the smooth muscle responds. At any age, Green's data shows improvement in 8 to 12 weeks. The endothelium upregulates nitric oxide synthase within days of beginning regular exercise. Breathing retraining normalizes carbon dioxide levels within weeks. Parasympathetic activation from relaxation practices reduces sympathetic tone measurably within a single session. The muscle you never trained is not dead, it is waiting. It is waiting for the inputs it was designed to receive. Movement, proper breathing, rest. The same inputs your grandmother's generation received without thinking about them because their lives demanded movement, because they breathed properly, because they were not hunched over screens, because their stress was acute and episodic rather than chronic and unrelenting. The practical prescription is almost embarrassingly simple. Walk briskly for 30 minutes most days. This provides the sheer stress stimulus. Breathe slowly through your nose at rest, 5 to 6 breaths per minute when you can manage it. This maintains carbon dioxide levels that keep smooth muscle relaxed. Find 10 minutes daily for genuine parasympathetic activation, whether that is meditation or prayer or sitting quietly in a garden or whatever allows your nervous system to shift out of sympathetic dominance. These three inputs, movement, breath, and calm, are the training program for the muscle you never knew you had. They are not optional lifestyle enhancements. They are the mechanical and chemical inputs that determine whether your vascular smooth muscle maintains its contractile phenotype or shifts to the synthetic phenotype that stiffens your arteries and ages your organs. Your grandmother never said, "Train your vascular smooth muscle." She said, "Go for a walk. Take a deep breath. Sit down and rest for a minute." She said, "Stop worrying so much." She said, "Slow down." She was describing the three inputs to vascular smooth muscle health in the language of common sense. Movement for shear stress, deep breathing for carbon dioxide, rest for parasympathetic activation. The muscle that controls how fast you age was being trained by the life she lived without ever knowing it existed, the life that modern convenience has engineered away, the life you can choose to rebuild, one walk, one breath, one quiet moment at a time. The muscle is waiting. It has always been waiting. It does not care how old you are. It cares whether the signal arrives.